Hi everyone, welcome back to Useful Genetics. This is lecture 2D. We're going to talk about two ways that mutations that are in coding sequences may nevertheless not change the function of the protein they specify. The first is that the mutation may be silent, that is, it doesn't change the amino acid that it specifies. The second is that the mutation may be neutral. It changes the amino acid, but that change does not interfere with the protein's normal function. And now, in the previous lecture, we talked about why most mutations are neutral because they're in the very large fraction of the genome that either is not part of a gene or that is in the introns of genes. Now we're thinking about the 2% of the genome that does code for proteins. Even in here, many changes don't change an amino acid. Now, because this depends on the properties of the genetic code, I just wanted to take a minute to talk about how we display the genetic code, because it's not obvious if it's not explained. So you've seen, I know you've seen because I showed it in Module 1, displays of the genetic code like this, where there's this complicated table with letters on all sides. It's hard to figure out what's going on. Really, tables like this are just a more compact way of displaying a list. So you can consider each part of the table as being part of a list. So we have listing the entire genetic code in this list. That's easy to understand, but it's pretty inconvenient to represent in a picture on a page. And so it's usually assembled into this more clever table diagram. The basic structure of the information, though, is that the three bases on the left side are three bases that form a triplet in the messenger RNA. And they will be translated as instructions to put a particular amino acid at this position in the protein sequence. And the three letters on the right are the abbreviation of the name of the corresponding amino acid. In this case, GGG specifies glycine. Now, there are other ways to represent genetic codes. Molecular biologists kind of like making things clever. So, for instance, you can represent it as this complex circular diagram instead of a table. This is more elegant, but I don't find it easier to understand. Now, back to the question, why most mutations in coding sequences are neutral. And the first part is that many changes don't change an amino acid because the genetic code has the property of being highly redundant. Redundant means you've got more than you need. In this case, it means we've got more information than we need. The genetic code could, in principle, specify 64 different things because there are 64 different ways to put three amino acids, three bases together in different orders when you've got 4 different bases to choose from. But there's only 20 different amino acids. So the result of the evolution of the genetic code has been that most amino acids, such as valine, are specified by groups of codons. Here, 4 related codons, they differ in their third position, all specify valine. Many other amino acids also are specified by groups of 4 codons. Some are specified by 3 or 2 codons, or leucine, specified by 4 codons. Only a couple of amino acids have a unique codon specifying them. Now, what this means is that it's possible to change the codon, to change the triplet code in the net messenger RNA without changing the amino acid that's inserted. And that's shown diagrammatically here. So here's a schematic drawing of the effect of different mutations on which amino acid is inserted. The green line here represents a messenger RNA, and we've highlighted a particular codon, particular triplet, the base is GAC. which the genetic code table tells us instruct the ribosome to insert aspartate, the amino acid aspartate, at this position in the growing chain. On the left, we've got a transfer RNA. In particular, we've got the transfer RNA that carries aspartate. On one end, it has 3 bases, the anticodon, that can form base pairs with the codon in the messenger RNA. And on the other end, it's loaded with the amino acid aspartate, ready to insert that into the growing protein chain. Now let's consider the consequences of mutations at this position. First, let's consider a mutation that does change an amino acid. So consider a mutation at this position, A here, that replaces the A with a C. Now, this mutation wouldn't have happened in the messenger RNA, it would have happened in the DNA, but the consequence is that a messenger RNA is made that contains a C instead of an A. What's going to happen? Well, we look at the genetic code table, and we see that the mutation has changed that codon into a codon that specifies alanine. This mutant messenger RNA, therefore, will not accept this transfer RNA at all because it can't form the correct base pairs in the anticodon. And so instead, it will form a codon-anticodon pairing with a different tRNA, one coding for alanine. Now, what about a mutation that is in a different place that's silent? So here we're considering a mutation at the G. It's replaced the C, with a U. Of course, in the DNA, that would be a T, but in the messenger RNA, that's a U. So now the new codon is GAU. Look up here, we've gone from GAC to GAU, but we haven't changed the amino acid that's specified. It's still aspartate. And in fact, this messenger RNA will still pair with the same transfer RNA because many transfer RNAs that code for amino acids with redundancy are actually, the physical manifestation of this redundancy is that the third position anticodon base pairs quite flexibly with the third position codon base. And in fact, the G of this transfer RNA will pair with a U as well as with a C. So that's one way that changes to coding sequences don't change function because many of them don't change the amino acid at all. The other way is that amino acids may be changed by a mutation, but this may not change the protein's function. This is seen when populations are examined. It's thought that at least half of the non-silent differences in DNA sequence between different people are actually functionally neutral. They don't change protein function. There's an example of this here. This is data from an analysis of a protein called SRC. This is a regulatory protein that regulates cell growth. And what these authors did was they created mutations at many different places in this protein, and then they tested them to see if they changed the function of the protein. And they put red and green dots onto the structure of the protein to show us where changes made a difference and where they didn't. All positions with red dots are positions where changing the amino acid changed the function of the protein, usually making it work less well. The green dots are positions where changes to the amino acids didn't change the function of the protein at all. Now, these are only the places that they tested. There would be many more places that did change function and many more places that didn't change function. So what we've done, we've started with what we already knew, and we've considered silent mutations that change the DNA sequence of a coding region but don't change the amino acid that's inserted, and neutral mutations where a different amino acid is inserted, but it doesn't change the protein's function. Now, There's still the section of the protein where changes make important differences that affect phenotype. We'll talk about these quite a bit in Module 3. But coming up next, we're going to think about a particular category of non-neutral changes in coding sequences, and that's the changes that are called frame shifts. I hope to see you there.